send it over to Kim.
Thanks, Todd. I met Michele the first time in 2019 at the very last in-person OSF convention. She received her Bachelor of Science from MIT in Massachusetts in biology and biomedical engineering. And then she got her Master's of Applied Science from the U of T here in Ontario in biomedical engineering. And she's currently pursuing her doctorate at the U of Guelph in epidemiology. For her master's thesis, she developed microfluidic devices to study the bacteria that cause Lyme disease. She's also a trained vet tech and practiced 5 years in both research and private clinics. Prior to pursuing her master's degree, she worked in healthcare as an account manager, selling diagnostic imaging equipment, including MRIs, CT, PET CT scanners. She's lived and traveled across much of the US, but for the past 10 years, she resides in Ontario. She loves working at the intersection of research and development and the application knowledge translation. Michele's current role leverages her technical skills in a niche area focused on public health issues impacting both animals and humans, and nowadays referred to as One Health. And for her, the best part has been the field work and getting to know the shepherds. And she, as part of her research, came out to my farm, and we had a wonderful day with her and her assistants. And I know another farm that she had came from, and the same thing. She says that farming keeps people young. So I will hand it over to Michele.
Thanks so much, Kim. I appreciate that introduction, and I just want to say I'm honored to be here and really excited. So I will share my screen.
Yep, and everything should be there to bring that up.
Oops. Okay.
Perfect.
Excellent. Okay. So let me just, I just wanted to mention that if you have a burning question that is very confusing with something I've presented, you can interrupt me, but otherwise, just to respect everyone's time tonight, if you want to type it in the chat and we can discuss it at the end. And I encourage you to ask your question just so that I can see faces and get to know you better rather than just looking at my face. So, all right. So, shrink this. Okay. So here to Cash a Killer, Cache Valley virus, to discuss Cache Valley virus in Ontario sheep. And it's proving to me to be quite difficult to cache this killer, considering it's quite obscure in terms of cases diagnosed, but omnipresent in terms of exposure. So tonight I'll be covering a variety of topics that I feel are important for you to be aware of in terms of trying to properly assess the risk of exposure to the virus and ultimately disease to yourself as well as your flocks. And so I will cover the virus transmission cycle, which is important considering this is a mosquito-borne disease. So there are many factors that need to be considered, and it's essentially like an optimization process. And once achieved, then disease manifests. I will also cover some of the clinical signs to look out for if you are thinking that one of your lambs might be infected with the virus. I'll then cover briefly diagnostics, because there are multiple tests that can be used to determine infection status, and you interpret those tests differently. I'll then cover historically what we know about Cache Valley virus prevalence in Canada and specifically in Ontario. We'll then cover risk factors in your sheep flocks, specifically in Ontario, both for exposure to the virus and disease. And then I'll touch upon my current research and share with you preliminary results and wrap things up with a summary slide. So first, a little trivia from the origins of the name. back in the early 19th century, fur expeditions were extremely common through this region, and the valley was subsequently used for annual gatherings of mountain men. Many of the trappers who worked in the valley came from Hudson's Bay Company, in fact, and the name Cache Valley was derived by the fur trappers who hid their trading goods in caches in that region. In the mid-1950s, during routine mosquito surveillance for other viruses known to cause disease in humans, they encountered this new viral strain, which they named based on the region where it was first identified, Cache Valley virus. Not until the late 1980s did they discover that the virus can actually cause disease in animals due to an outbreak in a sheep flock in Texas. Later on, they had conducted various exposure studies on a variety of wild animals as well as livestock, and realized that the virus can infect a wide variety of animals. It also can infect humans, although it's rarely reported. But again, the virus can only be transmitted through an infected mosquito bite. And then here's just dropped a pin of the location. It's actually a really beautiful region. So Cache Valley virus transmission cycle. Because it's mosquito-borne, there are actually many factors that we need to look at that can influence the exposure of the virus and either help or hinder its continuation in its natural cycle in the environment. And as I go through these next set of slides fleshing out this diagram, it'll help in guiding our understanding of why Cache Valley virus disease occurs in some years, but not others, and occurs in certain regions, but not other geographic regions. And it will reveal ultimately an optimization process by identifying factors that are necessary for disease and other factors that are essential, but on their own insufficient to cause Cache Valley virus disease. So first, a little bit about the virus. It's from the family Orthobunia viruses. There's actually hundreds across the globe, but only a small subset have been identified as causing disease in animals or humans. They are unique to certain geographic regions. In North America, there are 4 specific human and veterinary pathogens that we're aware of: Cache Valley virus, Jamestown Canyon virus, snowshoe hare virus, and La Crosse virus. The first 3 are endemic in Canada, as well as America and some parts of Mexico and further south. La Crosse virus, though, has not been reported yet in Canada. However, many cases have been reported in states that border the Canadian border, and the mosquito species that carry La Crosse virus are well established here in Southern Ontario. So it's really just a matter of time before we're able to detect this virus. These bottom 3, Jamestown, snowshoe hare, and La Crosse, cause encephalitis in humans specifically. They don't infect animals, but they have been researched much greater than Cache Valley virus. And so they have helped reveal a lot of information that we now know about Cache Valley virus disease. Also, it is now considered by Public Health Agency of Canada as a top concern among vector-borne diseases, because Cache Valley virus is zoonotic. Again, it infects humans as well as animals. There's currently no treatment and no vaccine for either humans or animals. And there's potential for geographic expansion of the virus due to the expansion of the mosquitoes as a result of climate change. And the reason why I point this out is because if it's on the radar of Public Health Agency of Canada, hopefully additional funding will be put forth towards researching this virus with an ultimate goal of developing a vaccine. But currently, we're not there yet. So first, we look at mosquitoes and piecing together this puzzle of the transmission cycle, because without mosquitoes, we really don't care about the virus because we don't have disease. And when the mosquito is infected with the virus, in fact, it can influence the feeding behavior and other properties of the mosquito. Also, mosquitoes are known to be very sensitive to their environment, and being able to manipulate these factors might be able to empower us to possibly reduce risk of exposure and disease. Mosquitoes, in order to survive, require blood meals, So that introduces us to the host in this cycle. The mosquitoes that we know transmit Cache Valley virus predominantly feed on white-tailed deer, but they also prefer alternate animals, particularly large species, such as humans and sheep. And this is how the virus is continued in the cycle. The final piece is looking at the environmental components that influence both mosquitoes and hosts. And for this presentation, we're focused on the role of climate change, as well as farm management practices. And I'm particularly interested with the latter because we might have direct, more immediate control over that in terms of mitigating exposure risk. And so this diagram identifies key pieces that need to be understood, because when this cycle is optimized, Cache Valley virus is a concern in terms of exposure and potentially disease. So the mosquitoes become infected with Cache Valley virus by one of three methods. The first and most common is through, it's called horizontal transmission, whereby an infected mosquito takes a blood meal from its preferred host, the white-tailed deer. And in just a couple of days, enough virus has replicated in the deer such that subsequent mosquitoes that land and take a blood meal will also pick up the virus and continue to spread it in its natural cycle. The second method of transmission is considered vertical transmission through a different generation, being passed on to the eggs, so transovarial transmission. So if an infected female lays eggs, she can pass on the virus to her progeny, both male and female mosquitoes. And that leads me to the third and final method for transmitting virus in a mosquito. For example, if a male mosquito reaches maturity and mates with a female, he can in turn pass on the virus to her. And then again, she can pass it on to her eggs, and the cycle continues. So oftentimes people don't consider male mosquitoes as a risk for the transmission cycle because they don't take blood meals. Rather, they get their nutrition from plant nectar. But as you can see here, they can pass it on through mating. So it's not the predominant method, but it's still something that we always need to keep in mind. So there are two types of hosts that the mosquitoes feed on. The reservoir hosts are white-tailed deer. They can maintain the virus as well as amplify it. This means that the virus can actually replicate to sufficient levels to then be passed on to other mosquitoes. You also have dead-end hosts, such as sheep and humans. So while we may show clinical signs and become infected, the virus cannot replicate sufficiently to then be passed on. So we, in fact, are considered diluting the virus in nature. So next, I'll cover some clinical signs. And for those of you that may have been unfortunate to experience Cache Valley virus disease in your flock, you might be familiar with the images in this slide. So these are illustrating some of the classic signs that we see in lambs infected with Cache Valley virus. In the red font, there's identify the classic signs. On the left image, we see an infected lamb born as a stillborn. And we see the distinct humped spine and fused joints. On the lower right image, it's an example of an infected lamb that was born live. And we see the very clear pattern of the twisted spine. There's also occasional mummification. Additionally, the brain of the lamb may be underdeveloped. Affected lambs are usually born at term, but either stillborn or die soon after birth. Lambs infected with the virus in very early pregnancy may die and be resorbed. Lambs infected in later pregnancy are born normal and in fact have antibodies to the virus, so they have long-term immunity. There have also been dystocias reported in dams that have infected lambs, and delayed delivery has been noted as well. So next I'm going to discuss diagnostics, because if you are conversing with your veterinarian, perhaps about a possible case, I just want you to be familiar with the terminology used, or if you're reading about it in social media or a news article. So there are two ways to diagnose. The first is through isolating the virus, and that tells us active infection. And there's a variety of sources that we can test from, either blood, cerebral spinal fluid, referred to as CSF, which is really just the fluid around the brain and spine, or a tissue biopsy, usually of the brain. And the test itself is actually determining the sequence of the tissue and trying to see if there's a map with a genetic bank online of various Cache Valley virus strains that have been reported and are recorded. And if there is a match, we know that the sample has the virus. Often you'll hear RT-PCR, which stands for reverse transcriptase polymerase chain reaction. And while this is a very sensitive test, there is a big limitation in that there's rapid viral clearance. So in a healthy animal that has a normal immune system, if they are infected with the virus, it'll be cleared from the system within just a few days. But in the case, say, with a fetus who hasn't yet mounted an immune system in the first trimester, they are susceptible. But as they further develop, despite the clinical damage, they will develop an immune system that ultimately clears the virus. And that's why it is so difficult to actually isolate the virus from a lamb born infected. So the alternate way is to look for antibodies, so neutralizing antibodies to Cache Valley virus, and that tells us past exposure. And you'll hear the term throughout my talk, seropositivity, and that means the serum is positive for antibodies against Cache Valley virus. The source that we test is similar, either blood or cerebral spinal fluid. Some of the tests you might hear are virus neutralization assay, such as PRNT or plaque reduction neutralization test. Another common method is the ELISA, which is an immunology-based antibody assay. The limitation here is that we cannot distinguish current from past infection. So if you have a lamb that was born and you think's infected and you test for antibodies, which are positive, you can definitively determine it was infected by Cache Valley virus only if you are certain the lamb has not taken a colostrum feeding first. However, if you test the dam for antibodies and it's positive, you cannot definitively determine if that lamb was, or the dam was infected during pregnancy or prior. But we use those seroprevalence testing to establish prevalence or exposure to the virus. And so next I'll share what we know about exposure in Canada and in Ontario specifically among sheep flocks. There's not a lot of literature out there, in fact, regarding Cache Valley virus specifically, especially in Canada. But there was a study that came out of Saskatchewan in 2013 that tested for antibodies to 50 flocks and found that 47 of those were positive, meaning that 47 had been exposed to the virus. Specifically, out of 130 sheep sampled, 84 of those were seropositive. So high prevalence of virus in Saskatchewan. In Quebec, the first isolation of the virus was from a flock in January 2013. And in Ontario, the first report of the virus was reported in 2 flocks independently in December of 2011. We also have some exposure data from 2010, which looked at 26 flocks and found 21 of them to be seropositive for Cache Valley virus. And then looking at specific outbreaks, it's scattered about. There was one I came across in 2020 in Saskatchewan. I've heard rumors of such in Manitoba as well. There were outbreaks among flocks in Quebec this past winter, as well as the previous winter, and a handful in Ontario that you probably know better than I do. The fact is that it's not a reportable disease, so veterinarians are not required to report this information. So oftentimes it's announced more locally. And as such, with this limited data, it's unclear still why outbreaks occur sporadically with no obvious pattern in terms of particular flock or sheep within flocks or particular geographic regions and even years. And in Ontario, it's unclear why there seems to be a rise in cases of Cache Valley virus disease recently, given the high prevalence of antibodies in flocks. It's possible that there might be a new strain of Cache Valley virus that's a bit more virulent, or the increased precipitation events that lead into mosquito season have facilitated an increase in mosquito populations, in turn resulting in greater number of Cache Valley virus cases. These are all things that I'm looking at through my research. And so this is data from that 2010 survey I mentioned. There were 26 flocks with a great geographic spread here across Southern Ontario. And you can see that the 5 flocks that reported no exposure, at least in the sampled sheep, were centralized in the central portion. And it's important to realize that just because the sheep were seronegative for antibodies, we can't definitively say there's no virus there. It's possible that it's just a lower exposure levels, or there were certain features about the ewe sampled that didn't properly represent the true exposures of the whole flock. It's hard to say. So what are the risk factors in sheep, specifically in Ontario, for both exposure to the virus and disease? So we know that sheep, basically as fetuses, are most vulnerable in their first trimester because they don't have an established immune system. So specifically the first 50 days of gestation. If a dam is infected for the first time when pregnant, not all fetuses within a litter will necessarily become infected. Exposure prior to pregnancy will result in long-term immunity, thereby protecting the fetus during pregnancy. It is difficult to diagnose infection prior to birth. We do see a consistent seasonal trend, specifically the virus peaks in mosquitoes in August and September, which manifests in disease in sheep in December and January. In terms of year-over-year patterns, it's really quite sporadic. So this image is to help overlap breeding schedule with risk. So we know that we've isolated the virus from mosquitoes as early as mid-July to as late as early October, with the peak levels, meaning the most virus in mosquitoes, being in August and September. So if ewes are bred during this time, the fetuses are in their first and second gestation, a vulnerable state. And if they're infected then, due to a 5-month gestation, this is why we see disease in December and January. So to clarify, lambs and non-pregnant sheep that become infected typically remain subclinical and develop lifelong immunity. However, for pregnant ewes that do not have the pre-existing antibodies to Cache Valley virus, infection during the first trimester may severely damage the developing fetus. Specifically, if infection occurs during the first 28 days of gestation, the embryos usually die and are resorbed. Infection between days 28 to about 48 result in fetal malformation. Usually these lambs are born at term, either stillborn or alive, with obvious malformations of the legs, spine, and brain. And beyond, as I mentioned earlier, that day 50 of gestation, based on literature doing experimental research, they concluded that the fetus will not be infected and born healthy with antibodies to the virus. So how do we know about this virulence period? Studies such as this classic one out of Connecticut, which conducted mosquito surveillance from May through October for 16 years, looking for the virus, identified it in 16 different mosquito species. All of these, except for one, are established in Ontario and helps us better anticipate the risks that we face here in Ontario. And as we can see, Cache Valley virus infects many mosquito species, but Cache Valley virus is almost uniformly detected in August and early autumn, regardless of the mosquito species. So while the abundance of the mosquitoes is declining, as we see in the black lines, the amount of virus identified by the red circles is on the rise beginning in late summer. So one more figure I wanted to pull from this study is just a nice representation of all the factors that we need to consider in terms of assessing exposure risk to the virus. So they looked at the different mosquitoes in terms of the number of sites across Connecticut, as well as the number of years they identified the virus, and finally the actual amount of virus from each mosquito species. And thinking back to that diagram I showed you earlier on with the virus transmission cycle, this is the type of data that we have to factor in when considering what influences the transmission cycle of virus. And specifically, we're wondering how many mosquitoes are present, both in absolute numbers and number of species, and where are they located? Are these locations the same as where sheep flocks reside or other animals that we're interested in terms of exposure risk? And what is the duration of mosquito populations for a given season? And what's the status of the virus in these populations? Because what we're interested in is relative to sheep, when and where are the infected mosquitoes located? So the final point I wanted to make in regards to risk factors has to do with environment. And so we know that mosquitoes are very sensitive to winter lows and summer highs, and that's kind of what helps identify where their habitat is established. Also, overall seasonal increases in precipitation facilitate populations of mosquitoes, as well as decreased number of frost days. And then land fragmentation, sometimes referred to as urban sprawl, can also impact both mosquito species and the host, the reservoir host, the white-tailed deer, whereby, say, logging occurs and agriculture spreads, or urban environments like residences spread into these regions that are the habitat of white-tailed deer and mosquito species that carry the virus. You're just increasing the risk of this overlap and perpetuating disease. And so how does this specifically translate to Cache Valley virus exposure? Well, let's consider the initial Cache Valley outbreak as a case study here. This occurred, as I mentioned in the beginning slides, in a research flock in Texas during the winter of 1986. That previous summer, in May and June, serum samples had been tested for antibodies to the virus, and only 5% of the flock was found to be seropositive, in other words, exposed to the virus. But in late winter, just after the outbreak, the flock was retested, and seropositivity had jumped to 63%. Interestingly, during the breeding season that preceded this awful outbreak of Cache Valley virus disease, the rainfall was nearly 4 times greater than the average over the previous 35 years. So this observation further supports the idea that within a mosquito season, the amount of Cache Valley virus is positively associated with rainfall. And specifically for disease transmission, the environment can actually, predicted climate change, especially here in Ontario, can extend the season for viral transmission. And it will increase the probability of overwintering survival of the mosquitoes that transmit Cache Valley virus. We also anticipate an increased range and abundance of both the mosquitoes and the virus. We also expect increased frequency of host vector overlap, again, due to the lag fragmentation. And climate change is actually capable, due to the warmer weather, of reducing time from infection to viral transmission. So if a mosquito becomes infected with the virus, if the temperature is rising, the virus can actually replicate quicker in the mosquito and be transmitted to another host more rapidly. So what I'm interested in is looking at whether farm management practices can offset risk of increased Cache Valley virus exposure posed by these anticipated changes to weather patterns. And so this leads me into my current research and preliminary results. I have 3 aims with my thesis, all focused on Ontario. The first is to identify associations between farm management practices and Cache Valley virus seropositive sheep, with the goal of decreasing risk of Cache Valley virus disease in sheep through farm management strategies. Second, I am identifying patterns of Cache Valley virus mosquito vectors in terms of abundance, seasonal trends, and geospatial clusters, with the goals of identifying hotspots for targeted Cache Valley virus surveillance and preventative education, building off of some of the work that I showed with you from those studies out of Connecticut. And then lastly, generating risk maps to highlight increased Cache Valley virus exposure for sheep and humans in the context of climate change over the 21st century, with the goal of guiding public health prevention and intervention efforts. But the results I'll share with you today are focused just on the first aim. So this past summer, I was able to conduct a field study, as Kim mentioned, comparing Cache Valley virus exposure in Ontario sheep to farm management practices. And I randomly identified and recruited 18 farms within 6 of the largest sheep districts and collected serum samples from 20 of the ewes on each of the farms. The eligibility was that the ewe had to have been on the farm for a full mosquito season prior to me being there and having given birth once. But those are the only criteria. And so these are the results. The orangish red circles indicate the flocks that were seropositive, so at least one ewe within the sample group had been exposed to the virus. And the X's identify the 2 flocks where all the ewes sampled were seronegative. So again, perhaps prevalence was low, or there were unique features of that sample group that didn't necessarily properly represent the full exposure, virus exposure of the flock. Overlapping my results with the results from 2010, hopefully you can see this all right. There's lots of synergy overall. And you can see a few locations where X is converted to circles. That could mean a couple of things, either that, again, it depends on features of the sheep sampled back in 2010 versus 2021, or perhaps the prevalence of the virus is rising. Also, I wanted to point out in this yellow circle that both times the ewes were seronegative. However, the farm has reported history of Cache Valley virus disease. So I mentioned that again, one of the limitations of exposure testing is that you can only determine that the sheep you tested were exposed or not, but it can't accurately tell you if there's virus in the area per se. And so in addition to collecting the serum samples, I also collected data from each of the shepherds, specifically on breeding schedule, flock movement and housing, particularly during the mosquito season, flock size, shearing schedule, surrounding environmental landscape, and any mosquito prevention practices, with the purpose of building models to determine whether there are associations between Cache Valley virus seropositive ewes and particular farm management practices. And so this figure is just a representation of the range in ages of the ewes that I sampled at each farm. And so we can see that overall there is quite a range from 1, which was actually just shy of 2, all the way up to, there was one that was 10 years old. And the red X's indicate the 2 farms where all the ewes sampled were seronegative. And interestingly, the age was consistent in all the ewes sampled at that time. So it's possible that those ewes were young enough and not yet had enough exposure time to be exposed to the virus if it's present on the farm. Or there's just no virus in the region where the sheep are located. We don't know, but this helps to start to analyze whether age, and specifically the number of mosquito season exposures, relate to Cache Valley virus seropositivity status. And so taking that a step further, I looked at the distribution of seroprevalence across flocks. So here, let's start with the first column with quote 0. Those are the 2 farms that had all ewes as seronegative, so no exposure to the virus. In the second column, there were 7 farms that had up at most a quarter of the ewes sampled positive for exposure to the virus. Moving along to the last column, we see 3 farms that at least 75% of the ewes sampled at each farm had been exposed to the virus. And so we can see that if the virus is present in the area that sheep frequent, there's still quite a variety in terms of percentage of those that are exposed versus those that are not. And so the takeaway from this is that if Cache Valley virus is identified near the farm, there's no guarantee of exposing all ewes to the virus by natural inoculation. So it's understandable why this practice is implemented. But this is telling us that if you know that you have virus on the farm, you've had cases in the past of the disease, and so you try to be preemptive and expose your ewes to those highly populated areas of mosquitoes prior to first breeding, you're still not guaranteed to have all of your ewes infected. So next, I looked at the association between the number of mosquito season exposures, so specifically that summer, fall exposure, and Cache Valley virus seropositivity. So again, starting with the first column on the left with the label 1 at the bottom, we see that of the ewes that I sampled that had only been exposed to 1 mosquito season prior to my arrival, less than 10% had been exposed to the virus. And then we see an upwards trend telling us that with more exposure time, they're likely to have antibodies to the virus. And then at the far end, with 9 and 10 seasons of exposure, all the ewes sampled were seropositive. They had been exposed to the virus. And so the takeaway with this image is that the more mosquito seasons a ewe has been exposed to, the more likely the ewe is to have Cache Valley virus antibodies. And logically, that makes sense.
Michelle, can I ask
Yes.
a question on that?
Absolutely.
So if your ewe lambs were being raised on grass prior to lambing, would that allow them to build up that immunity prior to having their first lambs?
That's a good question. I can't definitively say yet, but I understand where you're going. And the last set of slides, I do look at the movement in terms of where they are spending their time, because it would be nice to say if they're spending their time on grass or in a barn, are they less likely or more likely to be exposed? That is one of the ultimate goals. And so if you bear with me a little bit further, I'll show you that slide. So it's a great question, but I don't have a definitive answer yet.
Understandable.
So then I was looking at the at-risk population. So basically the ewes that were bred during mosquito season, because if they're not bred during mosquito season, we're not going to see disease and we're not interested. So 16 out of the 18 farms that I sampled from did breed their ewes during mosquito season. So among those, I was interested in looking at whether there was a relationship between when the ewes were first bred and history of Cache Valley virus disease in the flock. So here we see that for the flocks that bred their ewes for the first time at a year or younger, 75% of them reported history of Cache Valley virus disease on the farm. And for flocks that bred their ewes older than a year, 50% of them reported a history of Cache Valley virus disease on the farm. And so the takeaway with this figure is that if ewes are first bred when older than 1 year, it suggests it's less likely that the flock will experience Cache Valley virus disease. And then this is my final set of slides getting to that earlier question. I looked at the role of of housing. And so this figure, I just want you to notice the different sizes of the gray rectangles. And so what we're looking at here is lifetime exposure for each of the ewes I sampled during the summer and fall, during both daytime hours and nighttime hours. So for example, if I sampled from a 10-year-old ewe that spent her first year in total barn confinement and her remaining 9 years out on pasture, she would be represented here in the column barn and pasture, because I can't distinguish further than that. And so we see here that there's quite a variety in terms of where the sheep spend their daytime hours from farm to farm, as well as from daytime to nighttime. So then I looked at this through the lens of seropositivity, trying to see if there was a relationship between first day housing in the summer and fall and exposure to the virus. And from this figure, without having performed any kind of rigorous statistical analysis yet, which would distinguish just a random fluke from a pattern that we are confident will repeat, we can say that the ewes that spend time out in pasture or in the combination of barn, dry lot, and pasture had more exposure to the virus. But I will mention that in this particular slide, there was quite a variety in the sample size for the 5 locations. So further analysis really needs to be done, but the takeaway is that according to this figure, it suggests greater Cache Valley virus exposure among ewes that spend time in the pasture or in a combination of the barn, dry lot, and pasture. In the night, however, we have more equal sample sizes across the different housings. And so from here, we see that there's greater Cache Valley virus exposure among ewes that spend time in the barn and dry lot during the nighttime compared to the other 4 options: barn, pasture, barn and pasture, or the triple combination. And so just to summarize in a 1-pager of kind of everything about Cache Valley virus, take away from tonight, it is zoonotic, meaning it can infect both humans and animals, but only from an infected mosquito bite. Disease is primarily reported in sheep. Cache Valley virus is mosquito-borne and has been isolated from over 30 mosquito species that are established in Ontario. The virus exposure period is from July to early October, with the peak risk period being in August and September. Cache Valley virus causes congenital fetal deformities, with classic clinical signs being a curved or humped spine and fused joints. Disease exposure risk period is in the first 50 days of gestation. Infected sheep develop long-term immunity, which includes fetuses after that 50 days of gestation. Cache Valley virus is enzootic in North America. White-tailed deer are thought to be the main host, and there is an increased risk of disease if breeding during the Cache Valley virus season. So with that, I just want to emphasize that this was definitely a collaborative effort, and there are many that deserve a big thank you, including my thesis committee and my primary supervisor, Dr. Katie Clow, as well as a group of organizations I continuously working with: Public Health Ontario, Esri Canada, Sunnybrook Research Institute, tied with the University of Toronto, Public Health Agency of Canada, Sporometrics, of course, Ontario sheep farmers, and most importantly, all participating shepherds, because this research wouldn't exist without them. So a huge thank you. And with that, I'll take questions.
Yeah, I have a question here.
Yep.
So I was probably the most northern one that came back negative, but I
Okay.
just had a question about all the other flocks. Like my closest bush is about 1 kilometer away, and I spray with a Gita 1 to 2 times a week for flies. Does that, I don't know if anybody else has been spraying a Gita for flies and that, because that would kill mosquitoes too. Does that make a difference?
So there have been so few studies that have looked on the effectiveness of pesticides on mosquitoes. And, if they find one niche area, like a tree hole and a tree, say that's within usually around 1 kilometer of the farm that isn't impacted by the spraying, then you still have virus near the sheep. So my understanding is that it's just really challenging to maintain active pesticide for the mosquitoes. And there were several farms that did practice various things for flies, but there was not really a concentrated effort for the mosquitoes because they just are so omnipresent during the season here in Ontario. And so also, if you are trying to spray, you really have to cover the entire duration of the peak period, even as far out as, early October, or when you're breeding your sheep during that time.
Yeah, I'm just spraying for flies, so because I hate the flies.
Yeah, understandably.
We have one question here, a couple of questions here in the chat box. One, which was, are cattle not a factor or only sheep?
So cattle actually can be infected. In fact, it was one of the first cases that made researchers aware that the virus causes disease in animals. It's just not reported as often, possibly because it's not noticed or it's misdiagnosed, but more likely that it's just more common in sheep. But there is very little research that's been done on Cache Valley virus in cattle. Predominantly, it's just been focused on sheep, and mostly because a lot of the research stems from more familiar viruses like Acobani on the eastern side of the hemisphere, which we don't see here, and then also the Schmallenberg virus. But each virus has its own unique feature. Schmallenberg virus, which results in very similar clinical symptoms, is actually transmitted by the midge, the culicoides, the really tiny biting flies, as opposed to mosquitoes. And it's also not zoonotic, whereas Cache Valley virus is zoonotic. Why these viruses, all from the same family, are different in that regard is still not clear. But the short answer is it can affect cattle as well. It's just not as commonly reported.
Okay, and just moving on with some of the questions in the chat, could those seropositive cases be ones that aborted in early pregnancy?
So if they were seropositive, they would likely still have antibodies. So I'd be more inclined to think that those ewes were just quite young and hadn't had an opportunity to be exposed to the virus, especially considering that disease had been reported within the flock in the past.
I asked that question, and then I'm an ultrasound technician, and I was seeing abortions at 40-45 day pregnancies, and that's why I kind of asked that question, whether those ewes could have lost their pregnancies without even knowing it.
Oh,
and therefore
yes.
be positive on your testing without having known that they were pregnant.
Yes, that is true, because if you see that the fetus aborted prematurely, it would make sense, especially during mosquito season, to consider Cache Valley virus as the result. So if you were to then test the dam because you're wondering, and she's seronegative, then you know for sure it was not due to Cache Valley virus. If she's seropositive, then it's possible.
Perfect. And just moving on to next question in the chat. Do sheep inherit immunity? It seems like younger sheep, we see the issues.
Unfortunately, they don't. The antibodies that are required to protect the sheep are not passed on in the colostrum. So while sheep do get general important antibodies to protect them in the early stages of life, until they're properly exposed and mounting their own, that that will not protect against the virus. So sheep, the fetus is protected only if the dam is infected, but already has antibodies, because she can clear the virus quick enough such that it cannot cross the placenta to then infect the fetus. But otherwise, the only way to protect is by being directly infected. So that's why I was mentioning, interestingly, if a fetus is infected late in gestation, not only will they be born healthy, but they will have mounted an immune response and will have long-term antibodies when they're born.
Okay, and just moving on, another question that came in. We're all participating for, oh, yep.
Sorry to interrupt, just because my question's exactly on that comment she just made. So just so that I'm
Go ahead.
clear. Sorry to.
No.
So what you're saying then is if the ewe is infected or bitten after that 50 days, and it does cross the placental barrier at that point, that way the lamb will actually get protection, not through the colostrum like you said, but it'll be directly infected while still inside the ewe. Is that what you were saying?
Yes, because by that point, around that point, so it's not like absolute 50 days, but basically once the fetus has developed its own immune system, it's capable of clearing the virus. And that's the reason why even infected lambs, when they're born, the virus is gone because the immune system has cleared it, but the damage is already done.
Okay. So it's sort of a catch-22 then, in the sense that if your ewe has been infected before, she has her own immunity and will therefore clear the virus before it infects the lamb. So the lamb is safe that way.
But then at risk at birth.
If she hadn't been bitten and was infected after that 50 days, then both would be protected.
Yeah, exactly.
Good luck planning that one out.
Yeah.
Okay.
Yeah, exactly. But you nailed it. Yeah.
Thank you.
No problem. And the next question was, were all participating farms reporting past infections?
No. I have to look at the numbers. The best way I represented it was in that slide when I have that. Just give me one moment.
I had about 50%. I think I was in the 50% mark, Michelle, and I haven't had an infection or seen a lamb with Cache Valley.
And also, you tend to breed outside of the mosquito season. I think you started breeding typically just outside. So yeah, in fact, you were not represented in that figure as a result. Sorry, I'm getting distracted. But I did also want to mention that when I considered reporting history of disease on the farm, that actually included two groups. One, it was definitively diagnosed by a lab. And two, it included shepherds or veterinarians that didn't actually submit a sample, but based on clinical signs, were highly confident that it was Cache Valley. So it's possible that might have been including some farms that perhaps didn't have Cache Valley, but they thought they did. But again, I'm trying to, before I continue with the kind of rigorous quantitative analysis, I'm first trying to think about various trends to further flesh out. And so that's why I had presented that. Here we go. Okay, so there were, 3 out of 4 farms bred at 6 to 8 months of age, and 6 out of 8 farms bred between 9 and 12 months, and 2 out of 4 farms bred older than a year of age. And so of these In the first group, 3 out of 4 reported history. So I guess the short answer would just be 6 + 3, 9, 11 farms out of 18 report history, and 11 out of 16 that breed during the season report history of Cache Valley virus disease.
Good question, and thank you for the clarity on that one. There's another question here about how do the mosquitoes pick up the virus in the first place? Maybe a little bit of background on how it starts.
Sure. So essentially, the 3 ways that it can be passed on. Where it originally came from is kind of hard. They've looked at genetically sequencing the various strains. So the strain of virus that we see predominantly here in Ontario actually comes from Mexico. And how that occurred is not known. So it's predominantly, this strain took over around, I think it was around like 2010, from the strain that was first identified out in the Texas region. And so once you have the mosquitoes here, again, they can pass it on from biting animals while they're taking a blood meal, and then other mosquitoes that aren't infected feed off of that animal and pick up the virus. And if it's a female mosquito, she can pass it on to her progeny. And when the eggs hatch, they now have the virus. And so when the egg hatches and the mosquito is developing, the virus is also replicating. But as we see, we don't anticipate virus until late in the summer. So clearly it's taking a long time from when mosquito season starts before we actually are considering exposure risk, which tells us that there's a greater risk of virus exposure due to horizontal transmission. So infected mosquitoes feeding off of animals and spreading it that way versus passing it on through their eggs, because not all eggs will necessarily become infected, and not all infected eggs will necessarily make it to adulthood to then spread it during their blood meals or mating and spreading it that way. Does that help?
I'll keep an eye on the chat if there's any further question to that, or feel free to unmute yourself if you want to follow up on that question.
And one other,
what I was trying to figure out there is, so I understand all that process, but for example, where does it actually originate out there? So there's deer wandering around that are carrying the virus.
mm-hmm.
And there's sheep wandering around that are carrying the virus. So then once they are clinically positive, then if a mosquito bites them, are they then passing it on?
Good question. So a sheep is not a natural reservoir. So it's a dead end host, which means that once it's infected, the virus just can't replicate sufficiently in the bloodstream of the sheep, such that if other mosquitoes are feeding off the sheep, they can't pick up the virus. So you don't have to worry if you know that your sheep might have the virus. Mosquitoes feeding on them are not going to pass it on to other sheep. The deer, interestingly, when they are bitten, it's actually just a window of a few days when the virus is at such high levels and another mosquito, or maybe hundreds of mosquitoes, are feeding on that deer simultaneously, and they're picking up the virus. But after around 4 days, the virus is cleared from the deer system and the deer is now immune. So it's this really short window, but it's because there are so many deer around that allow the virus to perpetuate in its natural cycle.
Yeah, this year was a really wet fall, and the mosquitoes were very, very heavy. And I am in a heavily populated deer area as well, and I got hit with Cache Valley very badly this year. And so that's why I was trying, like, obviously it's come out of the deer.
Yeah.
But does this do the same kind of thing to the deer? as far as the
So-
deformities and all that kind of stuff? Like, how does it keep going there?
That's a good
Continue
question.
to affect us?
So there hasn't actually been research that has looked at whether deer have malformations, because the only way to know that for sure is that you experimentally have a deer infected and you observe the progress of the fetus through gestation, and that just hasn't been done. But the adults, as we know in sheep and deer, usually the symptoms are subclinical. So you wouldn't even know if the animal got bitten and was infected. And so their thoughts about, well, if you have a period, like a long dry spell, and so there are very few mosquitoes, and so now the animals like the sheep are no longer being exposed to the virus, and so fewer are actually immune, and then suddenly there's a heavy rain that basically results in an eruption of mosquitoes that have the virus. Now all of these vulnerable sheep are potentially going to be exposed, and if that's during breeding season, certainly you'll see a large number of cases of the disease. But this is a bit hypothetical, and that's one of the key goals of my research, is to overlap the climate data, the temperature and precipitation data, with what we know in terms of seroprevalence, to see if we can tease that out and definitively say exactly what weather patterns will suggest the following season is going to be bad for Cache Valley virus exposure.
Okay, yeah, that's kind of what I was
Yeah.
looking at, because definitely last fall was an excellent time for the mosquitoes, and this year's lambing was just horrendous for me. Cache Valley just hit me really bad. So that's what I was trying to figure out. So basically, I'm probably going to have to change my breeding around, and I'm 100% pasture. So again, because of that, then the sheep are out all night long, which again is when the mosquitoes are at their worst. So I can definitely see how the management of that, because I'm going to have to manage my breeding program around this. But now, hey, maybe all my sheep are infected and I don't need to worry again.
Right. I mean, you know, one of my dreams is coming up with a little snap test where you collect a drop of blood from a sheep and determine if it has antibodies. And if it does, then you don't have to test it again and you can breed without worrying. And if it does not, then you just need to be aware and maybe breed for a few seasons out of season or something. But it doesn't exist yet.
Okay. All right, great. Thank you very, very much.
And that's
Thanks.
actually a nice segue into our next question, which was a little bit about vaccine and what are your thoughts about something coming to stop lambs from being infected?
Yeah. Personally, I'm a huge proponent of it, but there's just not a lot of funding yet. I mean, we see what happens with funding in COVID vaccines. So that's why I was emphasizing that with Cache Valley virus becoming more or higher on the priority list for Public Health Agency of Canada, it will increase the likelihood. I've read about a few groups here and there, but nothing substantial in terms of advanced development for a vaccine. However, there have been a lot of improvements just in diagnosing these mosquito-borne viruses, even in humans, and that will increase attention and therefore funding. So even as late as till 2005 here in Canada, if a human showed clinical signs of viral encephalitis, if it wasn't due to equine encephalitis or it wasn't due to West Nile virus, it was usually never concluded the source of the problem, and they just tried to manage the various symptoms. But they radically improved their diagnostics for these cases as of 2005, and slowly the data is being shared. But you still have to be proactive if you think that it might be the cause of symptoms, insisting with your clinical provider to potentially test for various mosquito-borne viruses, because it's not commonly done, unless all the obvious risk factors are there, like you have mosquito bites or it's mosquito season. So.
Perfect. And Sean has a question about, excuse me, slide 47. How do those on farms compare as far as management, accelerated lambing versus annual, for example? And feel free to jump in, Sean.
Yeah, I was just wondering with the different age breeding groups there, like I was just wondering, just to generalize, if maybe the farms that are breeding after 12 months are, what you just missed, go back. Yeah,
I'm actually sorry, I'm cheating a bit because I wrote some notes underneath to remind myself about the breeding schedule for the farms. I know that the farms that actually were seronegative, they had and they breed less than a year, they have an accelerated program. The farms that bred later, after a year, they don't practice an accelerated breeding program.
I kind of wondered that just based on those, because that I'm accelerated as a bunch of the other folks are as well. So we tend to push and breed a little bit earlier than waiting for that full year, just based on available time.
Yeah,
is, right?
I completely understand. Yeah. But that's an interesting
Also, I
point.
guess, fall into that if you're breeding during the natural breeding season, that's basically just past the peak season for your mosquito infection anyway, right? So that would have an effect.
Yes. Yes, that's true.
Okay, and Jeff, we have a couple of questions from you in the chat box. Did you want to maybe unmute yourself and take them live?
Yeah, for sure. I was just looking to see, we've got some small statistical numbers, and I know that's a challenge during animal studies. Is there something we can do or measure that would kind of bump up your statistical ability and maybe even your funding to measure those things on sheep farms?
Well, one thing is that if we could do repeat testing, if you have two titers at least 3 weeks apart, you can get a better idea of when infection might have occurred, if it was active or long ago. Because when you are testing for antibodies, you get this number, and if it's a small number, you know one of two things, that either exposure was likely a long time ago, or it just happened and the immune system is in the process of creating these antibodies, but you can't distinguish. But if you have two tests a few weeks apart, then you can see the trend. Is it increasing? And then you know it's more active. In terms of numbers, one thing I am looking at is, so while I only have 18 farms, I do have 364 ewes to look at. And so that's why a lot of this, with these figures, it's more percentages that are being showed, but that helps bump up my sample size too. And I know when the ewes were specifically first bred in or out of season compared to their seropositivity status. So that does help.
Yeah, I hear you, and it's difficult to do the animal studies and get the statistical numbers that really kind of mark that as absolutes, right? I'm looking for, can we look at the mosquito population in the area and collect that to add to? Can we map the exposure?
Yep. So that's
and how do you find the funding for that? That's more where I'm at.
Yep. I'm dwindling with time, sadly, but my second objective, so I have all the surveillance data of mosquitoes captured in Ontario, graciously provided through the Public Health Ontario from 2002 to 2021. So I know the different mosquito species, how many were in each location each week for those 18 plus years. But we've been testing a subset for Cache Valley virus. And we looked at a few years where we thought we might have a good chance of identifying. And we tested 200 to 300 mosquitoes. And we, in all those years of testing, we only found one positive for Cache Valley virus, one mosquito pool. It's really looking for a needle in a haystack. And that was kind of near Bruce Peninsula. I can follow up. I'm sorry, actually, that was by St. Petersburg or Peterborough, was that one positive a few years back. And
well, you found your farm because I'm near Peterborough. So if you'd like to test our mosquitoes and our sheep, you're welcome.
Okay. Yeah, it's so I've been in touch with a group out of New York that does surveillance, and they actually even offer to assist with testing the mosquitoes, but you have to test thousands and thousands. The success rate is about 0.01% of the pools are positive for the virus. It's like minuscule and really hard to identify positives. So right now, while trying to continue that work of actually testing a subset of mosquitoes, I am also at least looking at trends in the different species to try to predict based on what has been observed in New York State and Connecticut State that has a lot of data on positive mosquitoes to try to anticipate trends that we'll see and identify hotspots throughout Ontario.
And just moving on, we've got a couple other questions in the chat box. How quickly do suspect abortions need to be sent to Guelph to receive good lab reports?
Well, the thing is, if it's a suspect, it's possible that it was infected months prior. And so you could test for the antibodies, in fact, actually, so I take that back. And then I guess if you think it's suspect, then you can ship it. But you definitely want to coordinate with the veterinarian to ensure you're getting the right samples. I believe here at Guelph, at the Animal Health Lab, they outsource the testing to a group in Texas that I worked with. And it's a trio of, I think it's cerebral spinal fluid, part of kidney biopsy. So basically you send the fetus and they collect the samples from there. And I have been trying to probe to see if there's a way to get financial assistance so that shepherds can submit this free of charge. But I haven't been successful yet, but I am trying.
This is the other Jeff from near Peterborough there. I had a question a little up earlier in the chat box. You said mummified abortions and stillborns can also be a sign, I guess. So they're not necessarily all deformities all the time then?
So I can't definitively say. I just know that while mummification has been a reported sign in a lamb infected with Cache Valley, it's also a sign in so many other types of infections or other causes of disease. Likewise with being stillborn or aborting prematurely, it can be a sign of Cache Valley, but it also can be a sign of so many other things. So if you get a mummified lamb, you don't know for sure that it's due to Cache Valley virus.
I guess we had probably about 50 users or so that had it in our December lambing, I guess. And I tend to notice we've had it in the past that sometimes you seem to get a few more stillborns and stuff along with the Cache Valley virus. And I just wondered if there's something common between the two things there, or whether they're related, I guess.
Yeah, it's certainly possible. And this is the Peterborough area too, you were mentioning?
Yeah. The vet says it can't be related, but I don't know. I've argued with the vet before, I guess. In a good way.
Yeah.
Okay, we've got two other questions here. I'm going to jump to the last one that just come in because I think the one before it might be a good wrap up discussion question. But from the discussions on the sheet page this year, I would suggest that infections happened earlier this year, October to December lambing. If this is true, could this become a risk for different lambing schedules?
In theory, yes, but so how early were the cases reported, and was that definitively determined?
This is Rebecca Parker again, the ultrasound technician. We had quite a long discussion on our land page in January, and a lot of people had infections through the late fall and into December and January. I saw a lot of abortion issues happening in October, so these are being infected in August, September, losing their pregnancies before they became viable, I guess. Which is indicating maybe a little bit earlier infections, not just our, possibly. Is it possible that these mosquitoes could be infective earlier in the season, therefore affecting pregnancies in the summer, etc.?
So I refer to this slide like all the time and count on my fingers, because if, say, a ewe became pregnant in August and became infected shortly thereafter, it would be expected that an abortion could occur even as early as October. Because
I'm asking this because we obviously have accelerated lambing in Ontario, and we have ewes that are pregnant potentially anytime in the summertime when mosquitoes could be around. Are mosquitoes only infective in the late year, or could they be infective earlier in the year?
Typically earlier in the year, there's just not enough virus in the mosquito to be passed on to the sheep.
Okay, that's kind of what I need to know. Thank you.
Okay, sure.
Okay, and then the last question that came in was one, I guess, more to the group, but you may have some comments as well about have others seen neurological effects? November hit us hard with Cache Valley virus, but January we didn't see any. And feel free, Steve, if you want to jump in on that one.
It's actually Lisa here.
Oh, hey, Lisa. But good question, but feel free to jump in on that one.
Yeah, so what we were seeing is not only the deformed lambs, but we were also seeing lambs that just didn't get going. Their heads were a little bigger than what they should have been compared to their bodies. And we were seeing a fair bit of that, and they did not survive. So what we're wondering is if anyone else was seeing things like that. Normally we would see Cache Valley in January group, but we haven't seen any in our January group, but we definitely saw them in the November group.
I too am curious what the group says, because I've read a couple cases where there's some sort of malformation in the skull of actually in goats that were positive for Cache Valley virus infection and like a shortened jaw. But
do you mean like a parrot mouth?
yes.
Yeah, we saw a couple of those as well.
Okay.
So I lost, like I mentioned before, I lost a number of lambs this year that were definitely to Cache Valley deformities, a lot of that kind of thing. I also found that for whatever reason, a lot of my lambs were not doing as well as they should. And I've had lambs that were slow and not keeping up, and lamb losses that I don't generally face. I haven't checked for mouths, but now that you've got a point, there might be a couple parrot mouths out there too, because we're thinking of some cute little faces that might be off. So there might be some connection there. Now mine have all been February losses though.
I know.
I guess we, well, I guess we had kind of seen it in all shapes and sizes. I don't know. We were just filling a wheelbarrow regularly. I guess you kind of start not paying as much attention, but it's just, we did have some slower lambs getting started, just all kinds of weird stuff. You get some that just come out with just one leg stiff that lamb couldn't get up because of it, or I don't know. It was just all kinds of different signs, I guess.
Yeah, and I'd have to say mine's been about the same this year too. It's been a very depressing year. The only thing that I can definitely peg is there's no question is Cache Valley. I haven't got any testing done, but you know, I've had to cut lambs out of ewes and everything else because of it. But yeah, the lamb losses have just been phenomenal, and a lot of it doesn't make sense. Out of ones that had appeared to be decent lambs or slower to start, or you know, the same kind of thing. It just a lot of them, and that's really hard when I only run 30 sheep flock.
Is there any discussion of having a reporting system among the small ruminant vets, so we can get a bit better idea of how many flocks are getting affected? It seemed like a lot of the flocks were affected this year.
I agree. I think there's a lot of value there. When I first started, I will say that I was told that it's not that common, but just in talking to shepherds, it's a lot more common than was originally conveyed to me. So I certainly think there's significant value in that.
Perfect. Well, I've just checked the chat box and I do not see any other questions. So maybe last call for any verbal questions or questions through the chat. And if not, I'll turn it back to over to Todd.
Can I just pose one question to the cases where there was a significant number in November and February? Out of curiosity, were those younger ewes, or like how old were they?
My older ewes, they lambed in January, and yeah, so a lot of mine would be my younger ewes. And the other thing is I still have 4 or 5 ewes that haven't even come in yet. So then there's also the possibility, have they already aborted and then rebred? So yeah, my older ewes, they've got perfectly fine lambs at their side. And I have a feeling I may have been hit by Cache Valley about 4 years ago too.
And where do they spend their time during the day and night? And
I'm 100% pasture, 100% pasture operation. And I live out in an area that is heavy deer population. I'm on the Cardin Plains, so it's also a lot of water in this area too. So excellent mosquito land.
Okay.
As I said, I think I'm going to change my breeding program around to help that I will only breed January ewes, will only be my older ewes after this discussion.
For us, our ewes would all been 2 years and older.
Okay.
Yeah, so the group that we had lambing there in November were mixed, ewe lambs and older girls. And it's the ewe lambs that got affected the worst.
Okay. It's like ideally in terms of modifying a breeding schedule, as long as they can be outside of that first 2 months of gestation by the time they hit mid-July.
And the challenge with that is you're affecting our highest priced lambs. So
I know.
Those lambs that were lost are the most valuable lambs you can make every year.
Trying to come up with better solutions.
Yeah, ours are pasture based as well. And really to have an idea to put them in the barn, that's not possible because there's other breeding groups in lambing at that time. So it's all part of the program.
Mhm.
And we're right in Greenock Swamp, so it's Mosquito Valley.
Mhm.
Perfect, and a bunch of thank yous and positive responses for the presentation tonight.
Still last quick comment, well, maybe not last, but a comment with going back to what Rebecca just said there. Do you think if we did have some sort of database type thing that would track or at least give you or whoever a bit of a guideline as to the prevalence, would that help your position as far as getting funding for further projects and that sort of thing?
Yeah, I think that'd be incredibly valuable. And I kind of have a short list of questions. It's really just, at what age do they start breeding and where do they spend their time during the day and night? It's like 4 questions. I've reduced it from my lengthy questionnaire that the poor shepherds were put through. And I passed that on actually to a vet in New York State because they're trying to collect data for similar purposes. So an active database that collects this would be incredibly valuable for me, for all shepherds, I think. So I really like this idea. Certainly we'll try to present it to the small ruminant veterinarian here at University of Guelph, but any momentum that can be generated on the end of Sheep Farmers of Ontario would be incredibly helpful.
I would think that shouldn't be something like maybe Todd or you guys would have something to say about. I think that should be something we could pull together on our own.
Sounds
yeah.
sounds like a resolution, doesn't it, Sean?
Yeah, I guess so.
And I can send the questions that I always wonder about, just like those 4 that I was curious, and anything that the shepherd wants to share is well received. But
Michelle, do you look at topography in your questionnaire? Because I mean, obviously, like we live in a high windy area and we touch wood so far, I've never had it. But a lot of the places we're having it are living in lower, wetland type areas. Do you put topography into your questionnaires?
I do. So I ask the shepherds if they're within about 1 kilometer of woodlot, marshlands, ponds, lakes, river, creek. But to complement that, I will be gathering kind of like objective data from a group out of University of Montreal that provides that raw data of temperature to try to overlap. But I am hoping to get land use, that's the word, land use too, because I imagine maybe there are situations where people aren't aware that there's a marsh within a couple kilometers of the farm. Not the shepherds I spoke to though, they seem to know everything very thoroughly, but I do think that's a very important factor as well.
Very good, Michelle. That was a fantastic presentation. And congratulations to the group as well. It's really great to have an engaged group and lots of really good questions and discussion to follow that up. I know you've taken time out of your personal time here this evening. I understand you've done this from the university due to internet challenges, which a lot of us have as well. So a big thank you from the group here from District 7 and from Ontario Sheep Farmers